So today we will learn the chapter 9. Chapter 9 is pathway that harvest chemical energies. Chapter 9 is composed of the 5 subjects, starting from the glucose oxidation, and also we will learn the in the presence of oxygen, so we will learn how glucose is oxidized. And finally, we will learn the metabolic pathways are interrelated and regulated. Okay, this is the first subject to say that there is the 3 catabolic process harvest energy from glucose. In previous chapter, we learned that what is catabolism. Catabolism means that highly ordered substance become to small unit. Okay, in our cell, usually use 3 catabolic process, starting from the glycolysis. Okay, so when there is oxygen, they will do the cellular respiration. Okay, or when there is no oxygen, fermentation will be done. It's like this. So there is the 3 catabolic processes harvest energy from glucose. In second slide, we will learn the basic concept, oxidation and reduction reaction. We call it as a Redox reactions. Okay? Oxidation means that loss of electrons, and reduction means that gain of electrons. So usually oxidation and reduction always occur together. For example, this A has the 2 electrons, and B, they don't have the electron. So during the redox reaction, A lost electron, A lost 2 electrons. So it means that this A is oxidized compound A. And B is they gained 2 electrons. Finally, B is reduced compound B is like this. The important thing is oxidation and reduction always occur together. So in this slide, we will learn the oxidation, reduction, and the energy. So figure 3.3 shows that oxidation, reduction, the energies. So starting from all of the molecule only has 1 carbon. For example, this is the methane. They have CH4, and this one is, we call it as, they have only 1 OH. This one we call it as methanol. After that, this is the oxygen. They have the double bond. This one we call it formaldehyde. After that, this H is changed to OH. We call it as formic. Finally, this is carbon oxide. So this is the most reduced status. After that, this is the most oxidized status. The important thing is that the more reduced a molecule, the more energy it has. It has stored in its covalent bond. For example, methane has this carbon. Between carbon and hydrogen, there is the covalent bond, covalent bond, covalent bond, covalent bond. And each CH bond, they have, each covalent bond, they have the 413 kilojoule per mole, but they have 1, 2, 3, 4. It means that total 1,600. However, the CO2, each C and O bond, they have 799 kilojoule per mole. But they only have 2, means that 1,500 is like this. It means that like this. The more reduced has the more, okay? Now in this slide, we will learn the NAD plus NADH. So NAD plus can be changed to NADH, or NADH can be changed to the NAD plus. These two molecules, we call it as electron carrier in reduction and oxidation redox reactions. First of all, NAD plus is electron acceptor. Okay, so after that, NAD plus is changed to NADH, is like this. Okay, if you see here, this is the NAD plus. So this is the, they have positive charges. So they receive the 1 hydrogen plus H plus, and they also receive the 2 electrons. After that, they will make the NADH, is like this. Okay, so this nitrogen and this carbon, they receive each electron. Okay, so NADH is the reduced form of NAD plus. So usually the NAD plus and 1 hydrogen and 2 electron, they will make the NADH like this. Okay, so in this slide, we will summarize the cellular respiration and the fermentation. So after glycolysis happens, glucose is changed to pyruvate. So when oxygen is present, okay, but when there is no oxygen, so when oxygen is available as the final electron acceptor, so there will be the 4 additional metabolic pathway, starting from the pyruvate oxidation and citric acid cycle. After that, there is the electron transport. Finally, there is the ATP synthesis. So we will learn the electron transport pathway starting from the complex 1 and complex 4, like this. The O2 oxygen is the final acceptor. O2 is function as the final electron acceptor. So it will change it to the H2O, is like this. So this process, we call it as the, so after glycolysis subsequent pathway, we call it as a cellular respiration. However, when there is no oxygen, fermentation happens. So it will make a lactate or alcohol like this. Okay. Now we will start the second subject. In the presence of oxygen, glucose is fully oxidized. Okay, let me start from the glycolysis like this. Okay, so glycolysis takes place in the cytoplasm. So our cell has like this. We have a nucleus and mitochondria like this. Glycolysis happened in the cytoplasm. Okay, here. Glycolysis happens here, okay, cytoplasm. So finally, it will convert, starting from the glucose. So it converts 1 molecule of glucose, finally to 2 molecule of pyruvate. Starting from the 6 carbon, finally it will make the strict 1, 2, 3 1, 2, 3. 3 carbon is like this. So usually in biochemistry class, we always we teach it that every step they are composed of step 10, but we always ask a student to memorize all of the pathway. But today is to very the basic concept. Okay, the glycolysis it changes glucose finally into the 2 molecule of pyruvate. So they are composed of the energy investing reaction, and also they are composed of energy harvesting reaction. From step 1 to step 5, we call it as the energy investing reaction. During the step 1, ATP is used, and also step 3, the ATP is used. So glucose is changed to glucose 1 phosphate. After 3 step is finished, so it will make the fructose 1,6 biphosphate. But today I will not teach you the details information. But important thing is that glucose is changed to fructose 1,6 biphosphate. So it means that 2 ATP is used. So during the step 1 to 5, it requires 1, 2 ATP. Okay, after energy reaction, O and H, this is we call it as aldehyde. The name of this molecule is the aldehyde 3-phosphate. So they will make the 2 molecules like this. So you don't have to memorize all of their structure. Okay, but important thing is energy investing reaction. So they require 2 ATP. Okay, so during the energy harvesting reaction, this glyceraldehyde 3-phosphate is changed to 2 molecule of glyceraldehyde 3-phosphate is changed to 2 molecule of pyruvate. So during this step, 2 NADH is synthesized, and also 1, 2, 3, 4. So each pathway they produce 2 ATP and 2 ATP. Finally, they will make the 4 ATPs. Okay, so during the energy harvesting reaction is composed of the step 6 to step 10, but they will make the 2 NADH and the 4 ATP is like this. Especially, we will learn the step 6. So this is step 6, starting from the glyceraldehyde 3-phosphate. Okay. So this is glyceraldehyde 3-phosphate. Okay. So step 6 means that glyceraldehyde 3-phosphate will become the 1,3 biphosphoglycerate. Okay, it means that so this carbon and these 2 oxygen and this H, we call it as aldehyde. So we call it as aldehyde. So aldehyde has the higher energy status. For example, this one, they have they are carbon and oxygen, 2 oxygen. This status looks like the carbon dioxide. So it means that their initial energy is higher. Finally, their product is like this. So this is the exergonic reaction. Exergonic reaction means that energy is released. So during this step, energy is released trapped in the energy released. So several energy is released. So this energy is trapped in the reduction of NAD plus to NADH. So this is we call it as the step 6, especially the oxidation and reduction reaction. This carbon is oxidized, but NAD plus is reduced. So that's why we call it as oxidation and reduction reaction. Okay, so step 7 is the starting from the 1,3 biphosphate to 3-phosphoglycerate. Okay, for example, this is the 1,3 biphosphoglycerate. So they have the phosphate like this. But this is the 3-phosphoglycerate. So phosphate is removed. Okay, this phosphate is removed. So during that time, 18.9 kilojoule, the energy is released. So this energy is trapped in the conversion of ADP to ATP. So this is, we call it as a substrate level phosphorylation. This step, we call it as the substrate level phosphorylation. So it happened in cytoplasm. So substrate level phosphorylation, we call it in Korean, we call it as a 기질 수준의 인산화. Okay, it happens in here, cytoplasm. Also, especially, we call it as a substrate level phosphorylation. Okay, so ADP is converted to the ATP. Okay, because this phosphate is removed. So during that time, 18.9 kilojoule is released, energy is released. This energy is trapped in the conversion of ADP to ATP. Okay. Next, so this is the glycolysis. Okay. So usually, during advanced subject, I will teach you the specific details information, but today I will give you the basic concept. Okay. Energy investing, energy harvesting. And also special step 6 and step 7 is like this. Okay. Okay, and let me go to the next step. After glycolysis is finished, okay, when there is oxygen is available, the pyruvate oxidation is happen. It's like this. So pyruvate oxidation happens in the mitochondrial matrix. So pyruvate is made. So after finished glycolysis, 2 molecules of pyruvate is synthesized. Okay. Okay, this pyruvate is transported into the mitochondria. So this pyruvate oxidation is happen in the mitochondrial matrix. Okay. So this pyruvate is oxidized. Okay. So this pyruvate, this pyruvate, they have this pyruvate is oxidized to CO2 and CH3C, or we call it as acetate. So this pyruvate, first this carboxyl group is changed to CO2, and this one will become the acetate. Okay, pyruvate is oxidized to the CO2 and acetate, and during that time NAD plus is reduced. Okay, reduced. After that, this acetate is bind to the coenzyme A, so they will make acetyl coenzyme A is like this. So like this, the carboxyl group is removed. Okay, so after that they will releasing CO2. Of that, NAD plus is reduced to the NADH. Simultaneously, coenzyme A bind to this acetate, so they will make the acetyl coenzyme A. So this is the pyruvate oxidation. Okay. So next step is citric acid cycle. Okay, we call it as citric acid cycle, starting from the acetyl coenzyme A. So after pyruvate oxidation happens, so they will make acetyl coenzyme A. After that, acetyl coenzyme A is starting point. It's like this. This is starting point. Okay. After that, there is the 8 step, starting from the step 1, step 2, step 3, step 4, 5, 6, 7, and 8 is like this. Okay. But today is the biology class, so I will not teach you the details, but I will teach you some important concept. Okay. Okay. So acetyl coenzyme A is, they have, how many carbon do they have? 유태현, how many carbon? 유태현, 너 불렀잖아. 저기 유태현, 김서진 옆에. How many carbon? 1, 2, 2. They have 2. So acetyl coenzyme A, so this is oxaloacetate. They have 4 carbon. 2 carbon and 4 carbon, and they combine to each other. So they will make the 6 carbon. So this is, we call it as a citrate. During the first step, it will make the citrate. That's why we call it as a citric acid cycle. Okay? This is why we call it as a citric acid cycle. Okay? So acetyl coenzyme is starting point. This is acetyl coenzyme A. So this acetate group is the oxidized. Each acetate group is oxidized. Finally, step 3 and step 4, this carbon will oxidize. It will make the CO2, and this carbonyl group also further oxidized, so it will make the CO2. So citric acid cycle, they will oxidize this acetyl group, and they will make the 2 molecules of CO2. So they are oxidized like this. So it means that acetyl coenzyme A, starting from the formaldehyde status, and they will make the carbon oxide. So it means that this is exergonic. So during that time, energy is released. Okay, so energy is captured by the GDP. So GDP will change it to GTP. And also NAD plus is reduced to NADH, and NADH. 3 molecules of NADH NAD plus is reduced to NADH. And also there is another Fadh plus is reduced to FADH2 like this. So energy is released is captured by GDP, NAD plus, and this is FAD plus. There is typo, this D plus, NAD plus, it's like this. Okay, the important thing is that what is starting point? Acetyl coenzyme A. You don't have to memorize the structure, but you have to know the acetyl coenzyme A. They have 2 carbons. So it binds to the oxaloacetate, 4 carbon. So finally, it will make the 6 carbon citrate. It's like this. So also, they will make the 2 carbon oxide, and 1 GDP is changed to 1 GTP, and 3 NAD plus is changed to 3 NADH, and 1 FAD plus we change it 1 FADH2. It's like this. For example, GTP, this is structure of GTP and ATP. They are nearly similar to each other. So usually the GTP can be easily changed to ATP. There are only one. There are two locations is different. So GTP is easily changed to ATP. Okay, so they have the same energy. Okay. After that, this is the citric acid cycle close-up, the final status. Okay. Okay. So this part C is that I will cover step 8. So this is step 8, the final cycle of citric acid cycle. So this is 4 carbon. This is, we call it as a malate, 1, 2, 3, 4 carbon. So this one will change it to oxaloacetate. So this status is further oxidized, so they will make like this. Okay, so this OH is changed to like this. It means that they are further oxidized. It means that their reaction is exergonic. So exergonic means that energy is released. So this energy is the trapped in the reduction of NAD plus to NADH is like this. Okay. At that, this oxaloacetate is bind to the acetyl coenzyme A, so they will make the citrate is like this. Okay. So during glycolysis, okay, during glycolysis, go see here. So during glycolysis, 2 ATP is used. However, 4 ATP is synthesized. So it means that 4 minus 2, how many ATP is synthesized? How many? 김도현? How many? 2. Minus 2 plus 4 means the total 2. So 2 ATP is synthesized. Okay, so after glycolysis, 1 molecule of glucose is changed to 2 molecules of pyruvate. Okay, so during the pyruvate oxidation, each pyruvate oxidation, CO2 is synthesized, and also it will make the NADH. 2 NADH is synthesized. So during glycolysis, 2 NADH is synthesized and 2 ATP is synthesized. So during the pyruvate oxidation, 2 NADH and 2 CO2 is synthesized. After that, there is a 2 citric acid cycle. So it means that each citric acid cycle synthesizes 2 CO2, 2 CO2, 2 CO2. Finally, they will make the 4 CO2. Okay, so each citric acid cycle, they will make the 1 GTP, 1 GTP. So it means that 2 GTP is changed to 2 ATP. So they will synthesize 2 ATP. Also, during the each citric acid cycle, they will make the 3 NADH. It means that 3, total 6. 1 FADH, 1 FADH, they will make the 2 FADH2. Total, so 1 molecule of glucose is 1, they have 6 carbon. So this 6 carbon is changed to 6 CO2. Each CO2 has 1 carbon. 6 CO2 means they have 6 carbon. After that, they will make the 2, 2, 6, 10 NADH. And they will make the 2 FADH2. Finally make the 4 ATP is like this. Okay. So actually in our textbook, they only have this one like this, but it is hard to understand, so I will make it like this. So I made this picture. Okay? Okay? We have to know. Okay, important thing is the 2 ATP, 2 ATP, 4 ATP. 6 carbon is changed to 6 CO2. And we have to memorize 10 NADH, 2 FADH2, it's like this. Okay, after that, now we will cover oxidative phosphorylation, this step. Oxidative phosphorylation step, we call it, they are composed of electron transport and also ATP synthesis. Another name of ATP synthesis, we call it as chemiosmosis. Okay, this process, we call it as oxidative phosphorylation. Okay, so oxidative phosphorylation means that in Korean, we call it as the 산화적 인산화. So during the oxidative phosphorylation, additional ATP is synthesized. Okay, so NAD plus is reduced to NADH, and FAD plus is reduced to FADH2. This is electron carrier. Okay, these 2 electron carrier is reoxidized. So it will change it to NAD plus and H plus. So this one will be changed, reoxidized to FAD plus, and 2H plus is like this. Okay, so and this is electron transporter system, and this is we synthesize the ATP. So this one, we call it as ATP synthesis, or another name is chemiosmosis. These 2 steps, totally we call it as oxidative phosphorylation. Oxidative phosphorylation. Okay, so let me see some details. So this is electron transport starting from the complex 1, complex 2, complex 3, and complex 4. Nowadays, some scientists find that the complex 4 is a very important role in the aging. So who studied this one? Complex 4? It's me. I studied complex 4. Okay? Okay? So if you are interested in complex 4, please let me know, okay? Okay? So So I published in aging cell. So I found that the complex pore is very important role in the aging. Okay. Okay. So they are electron transport. They are composed of the complex 1, and also there is a ubiquinone, complex 2, complex 3, and cytochrome C, and complex 4 is like this. Okay, so usually in advanced class, you have to memorize all of these steps. But in the biology course, the only thing we have to know is that, so this is the x-axis, this is the y-axis, is the free energy relative to oxygen. The oxygen is a final electron acceptor. Okay, starting from the NADH, so they have the 40 kilocalorie is higher than oxygen. How about the FADH2? So they have around the 40 calorie per mole is higher than oxygen. Okay, the important thing is that NADH and FADH is oxidized. They lost hydrogen like this. So it will make the NADH is the NAD plus, H plus. FADH2 is changed to FAD and plus 2H plus is like this. Yes, and also oxygen is the final electron acceptor. Okay, starting from the complex 1, so NADH is oxidized, and complex 2 is FADH2 is oxidized, but you don't have to memorize it. Okay, the important thing is that Professor Park is identified complex pore is important role in the aging. Okay, okay, but when you have to know the oxygen is a final electron acceptor. Okay, so this is like this, starting from the complex 1 and complex 2, complex 3, and complex 4. Okay, when in complex 1, NADH is changed to NAD plus and H plus. During that time, H plus, we call it as a proton. So this proton is actively transported to the intermembrane spaces. This is mitochondria. They are composed of the outer membrane and inner membrane. So these spaces, we call it as intermembrane spaces. So this proton is actively transported to these intermembrane spaces during electron transport. Also, complex 3 is also transport proton, and complex 4 also transport protons like this. So it means that intermembrane spaces, they have the higher proton concentration. But proton is their plus charges, so it creates concentration gradient. They are higher H plus gradient, and also they have higher positive charges like this. So it will make the proton motive pores. So this proton intermembrane spaces, they have the higher proton. So this proton is diffused to the mitochondrial matrix. This is mitochondrial cytoplasm. So diffusion of proton back to the mitochondrial matrix. During that time, it will make the ATP. They will synthesize the ATP. So this one, that's why we call it as chemiosmosis. H plus is chemiosmosis. They are higher osmosis like this. Okay. And you can see that if you see some detailed information here, so this intermembrane species, they have a higher proton. Okay, this proton is diffused through the channel. So potential energy, this potential energy is converted to kinetic energy. So they have the F1 subunit. So these polypeptides rotate. So this potential energy is changed to kinetic energy. So the energy transmitted to the F1 subunit. So it will activate this subunit. So they will make the ATP is like this. Who studied the ATP synthase? Okay. So I published this one in Nature Chemical Biology. So they are same. I identified that similar mechanism like this. So I identify some molecule. So they control this activity. Okay. This one I identified it. Okay. Nature Chemical Biology. Okay. Okay. So important thing is that ATP synthase, you don't have to know that ATP synthase is composed of F0 subunit. This unit is anchored in the membrane. You don't have to memorize it. But they have F1 subunit. So this is that they have catalytic effect, catalytic activity. So it will rotate it. So finally, it will change the ADP to ATP. Like this, okay? Okay, so let me oxidative phosphorylation. Okay, we finished. So we finished it, electron transport and the ATP synthase. Okay, synthesis mechanism. Okay. Now we will study the glycolysis and fermentation. Subject 4. So in the absence of oxygen, when there is no oxygen, even though there is no oxygen, some ATP can be made by glycolysis and fermentation. So these two steps occurs in the cytoplasm. So glycolysis occurs in cytoplasm, but from pyruvate oxidation, citric acid cycle, electron transport ATP synthesis occurs in where? In midterm exam, you have to know where. Mitochondria, okay? But fermentation occurs in the cytoplasm. So during the fermentation, it will make lactate, or alcohol is like this. Okay, let me see lactic acid fermentation. Okay, whenever we did whenever we did a lot of exercise, next day we have the stick in the muscle because lactic acid is produced. Because we did highly exercise in the muscle, there is not enough oxygen. That's why the lactic acid fermentation is happening in our muscle. In next day, we have a little bit okay. So lactic acid fermentation. So during glycolysis, one molecule of glucose is changed to 2-pyruvate. We already know that. During that time, 2 ATP is synthesized and 2 NADH is synthesized. Okay? And this pyruvate is changed to lactate. So this is the lactate structure. Okay? So this is oxidized form, and this is the reduced form. Okay? So at that time, lactate dehydrogenase. Lactate dehydrogenase function. So it will catalyze the lactic acid fermentation. So use NAD plus as a reducing agent. After that, it will reduce pyruvate to lactate. That during that time, NAD plus pyruvate. Okay. So whenever we did highly exercised, it means that our muscle don't have enough oxygen. At that time, fermentation, lactic acid fermentation is happen. So pyruvate changes to lactate. So during that time, NADH is used. During glycolysis, 2 NADH is produced, but during lactic acid fermentation, 2 NADH is used. It means that summary of glycolysis and lactic acid fermentation, there is only 2 ATP is synthesized. Okay? So let me go to the alcoholic fermentation. Okay? So alcoholic fermentation. Okay? Okay, so usually whenever we make the alcohol, we use the alcoholic fermentation. Okay? Pyruvate can be changed to 2-acetaldehyde, and in the acetaldehyde is changed to ethanol, is like this. So at that time, there is 2 enzyme is involved. You don't have to memorize it. First one is pyruvate decarboxylase. Decarboxyl. So during that time, CO2 is removed. So it will make this CO2 is removed. So it will make the acetaldehyde is like this. So finally, acetaldehyde is changed to ethanol. At that time, also NADH. They used NADH as a reducing agent. It will reduce acetaldehyde to the ethanol. During that time, starts regenerating NAD+. Okay, so during glycolysis, we produced 2 ATP and 2 NADH, but this 2 NADH is one glucose, we change it to 2 ethanol, but there is only 2 ATP in. You have to know the 2 ATP is synthesized. Okay? Okay? So let me summarize glycolysis and the cellular respiration. And next slide, I will summarize glycolysis and fermentation. Okay? So this is the summary. So during glycolysis and cellular respiration, 2 ATP, total 4 ATP is synthesized. Okay, and also 10 NADH and 2 FADH is synthesized. Okay, previously we learned that NADH, they have higher free energy compared to the O2. So usually the one NADH produced 2.5 ATP. Because we synthesized 10 NADH, it means that they will make total 25 ATP. How about FADH? So FADH has their lower free energy. Okay, one FADH2 produces 1.5 ATP. It means that 1.5 multiplied 2, they will make the total 3 ATP. It means that 4 plus 25 plus 3, total 32. So they will make the total 32 ATP. We have to memorize it. However, glycolysis and fermentation process, only 2 ATP, total 2 ATP is synthesized. Okay? During glycolysis, 2 NADH is synthesized, but it usually used, it always used like this. Okay? That's why the only total 2 ATP is synthesized. Okay? Okay. So in this slide, I will explain the fifth subject, this metabolic related and regulated. Okay, we learned that glycolysis, one glucose is changed to 2 pyruvate, and pyruvate oxidation happens, so it will make the acetyl coenzyme A. After citric acid cycle, that is the electron transport and ATP synthesis. But usually, glucose is main source, but sometimes several polysaccharides, and also some lipid, and also polysaccharide, or another protein, also broken. They are broken into the small subunit. That's why we call it as catabolic interconversion. So this one is also changed. Okay, so glycerol is changed from the glyceraldehyde 3-phosphate. So it will become the member of the glycolysis. Is like this. So usually the important thing is that several polysaccharides, lipid, and protein can all be broken down to provide energies. So why? The only ATP synthesis pathway is starting from the glycolysis to pyruvate and citric acid, electron transport, and ATP synthesis. This one pathway. But we also have lots of lipid, lots of polysaccharide, but this one can be changed like this. So it will become another component. Okay. Okay, and also we will learn the anabolic interconversion. Anabolic means that small subunit can become the highly ordered substrate. So this is glycolysis starting from the glucose to pyruvate. Gluconeogenesis means that starting from the pyruvate, it can make the glucose is like this. So this process, we call it as gluconeogenesis. So gluconeogenesis, they will make the glucose. That's why we call it as anabolic interconversion. Also, this acetyl coenzyme A can be used to make the fatty acid, and also another alpha-ketoglutarate. This one changes some amino acid, and after that it will change it to the purine base. Purine base is composed of adenine and guanine. And some oxaloacetate, first of all, it changes some amino acid. Finally, this one will change it to pyrimidine base, which is composed of thymine, cytosine, and uracil is like this. Okay, it means that metabolic pathway is interrelated and regulated. Okay, catabolic interconversion and anabolic interconversion. Okay. Okay, so today we learned pathway that harvest chemical energies. It's very difficult, so this one summarized everything. One, during glycolysis happens. When there is the oxygen, cellular respiration, so they will make the 32 ATP. When there is no oxygen, so they will make their fermentation. So they will make the only 2 ATP is like this. Okay, and also each catabolic interconversion and anabolic interconversion is always happen. Okay. Okay. So do you have any